So it's my pleasure to introduce this session. This session will focus on approaches and methods used in the development of new gene and cell-based therapies. We've got a fantastic panel of speakers who are going to cover a diverse range of topics, ranging from the use of CRISPR-Cas technology for targeted gene editing to phenotypic drug discovery and chemical genomics. So without any delay, I'll turn it over to Anne to introduce the speakers.
All right, so it's a real pleasure to introduce our first speaker, Dr. Xiang-Dong Fu, who's professor of cellular molecular medicine at UCSD. So Fu is well known for seminal contributions and discoveries in the field of RNA splicing. And Luke and I first thought about Fu for this session in particular because of his creative use of high-throughput technologies over the years. So Fu's studies focus on RNA regulation in a diversity of biological settings, including cellular reprogramming, which would have been another great story for today, neurodegenerative disease, and cancer. And so today he's going to tell us a new story, focus on new mechanistic insights into leukemia caused by mutations in splicing factors. So, Fu, thank you.
Oh, it's on. Thanks a lot, Anne and Luke. I'm very grateful to the invitation, and I think it's an honor to talk in your own home institutions, and also make you feel nervous a little bit. So I think it's a little bit a different crowd than I normally give a talk, and in this one I try to include more disease relevance in addition to our normal emphasis on mechanistic insight. But anyway, I try to blend both topics together. So talking about splicing disease, I think this is one of the, I would consider relevant to our research, one of the major advances in recent years out of genome sequencing. What's unexpected is we and many study gene regulations at the transcriptional and post-transcriptional levels love to have connections to disease so that we can justify funding from NIH. But for many years, we have been playing our little games in our own favorite factors, and without knowing there are any relevance to disease. But all of a sudden, in these publications, actually there are a few prior to this, and they found that in this particular pre-leukemia called myelodysplasia syndromes, MDS in short, that there were multiple very high-frequency mutations in multiple splicing factors, including particularly like this one, SRSF2 is the one I cloned when I was a postdoc, and it's the first trans-acting factors required for both constitutive and regulated splicing. And then so in various disease leukemia state, and even in AML, and this is one of the most prominent mutations found. And what's most interesting is that all the mutations found in one spot and change one amino acid in thousands of patients. So it's very curious how those things functions and at what levels in order to develop a therapy. So this is kind of a summarize what we learned. All those things with a kind of light indicating that factors contain mutations identified in the MDS patient. Okay, there are many of those things by direct sequencing. And what's interesting here is that all of them function based on the biochemistry of prior work, function in definition of a 3' side. So that naturally lead a hypothesis everybody favored, including the authors, that these things may cause disease by affecting some common target in splicing. Okay, it's very natural, and in fact, this is what has been focused on by the entire community thus far. Okay, just bear that in mind. But in studying this kind of disease mechanisms, one has to bear some key factors here, facts. One is that the MDS patient is a precancerous state. It's not a final cancer. So they are actually categorized by a poor proliferation state and a poor differentiation, so that a lot of cells get arrested at the embryonic stage and kind of near the progenitor cell, early progenitor cell stage. And MDS patient frequently transit to AML. This is acute myeloid form. It's more advanced disease. So that means that initially it shows some kind of inhibition to growth or proliferation, and eventually burst into rapid proliferation and differentiation. Okay, so this is resemble, in fact, in many aspects to the development of the solid tumor, that you started with a pre-oncogenic event and then synergized with a second hit or multiple other hits to become a real tumor. And one important thing which has been puzzled us for a long time at the field is that the leukemia in general has the lowest mutation rate than other tumors. Like melanoma, you get tons of those mutations. Most of them, of course, is passage mutations resulting from genome instability, and the genome instability is a common feature of cancer, but leukemia in general has a very low mutation rate. Okay, just keep that in mind why this is relevant for the rest of the talk. Okay, this function, there are many mutations identified, and then this one has been well characterized by using lock-in mice. Okay, so this is derived from this wonderful work published a year ago in Cancer Cell. In wild-type mice, you can see there's a normal bone marrow productions, and when you have a lock-in heterozygous, okay, you cannot tolerate homozygous lock-in, the mouse. Okay, then you see this leukopenia and anemia and other dysplasia phenotype, basically with a myeloid-skewed cell population. And in contrast, the total loss of these factors, SR, SF2, basically reduce the cell number in the general way and not cause abiotic lethal in the early state. So that the mutation is completely different than just loss of function of the gene. Okay, so whether it's a gain or altered function is a separate question, but clearly this is a lot, the mutation wipe out the function of the protein. Okay, and then these mutations, in fact, these factors has been well characterized at biochemical level to bind exonic regions to promote splice site selection. And such a binding has shown such a preference of a GGNG or CCNG, in fact, very degenerate. And then by categorizing the mutant protein for its binding preference, in fact, it maintains binding affinity to one class of site while showed reduced affinity to another class, so that is a kind of a tilting for splice site sequence recognition, but not completely lost. But this one is a complete loss. As a result of this splicing change, one of the key factors identified is EZH2, a polyclone component that has been shown to contribute to the disease phenotype in these studies. And ever since in vivo, overall, the study established the causal effect of the mutations in this particular gene. Okay, so that's very important and thorough studies. And we wish to study, because this mutations is heterozygous mutations. And the mutations, when you long-term introduce in a stable way in the animal models, sometimes you may miss some acute phenotype you fail to capture. So we choose to use quote-unquote a little bit artificial, or in a better way to say is more a reductionist approach to see how this mutation cause any phenotype or molecular event that we can capture, and then later on we worry about biological relevance. So this is our rationale in design, which is to construct this expression vector that drive your either wild type or mutant genes, HA tagged, from a tetracycline promoter. And then this transcriptional unit has a 3' UTR in which we build in shRNA against its own gene. So when you infect these things selected with a puromycin selection marker driven by different promoter, then you ended up replace endogenous gene with exogenous gene. So that way you can study wild type, compare wild type and the mutant. So now we introduce either wild type or mutant. This is a P95H. This is a protein 95 changed to histidine, and in fact there are multiple changes to lysine, arginine, and all produce this similar thing. I mean, as I said earlier, all the vast majority in this gene, the mutations happen in one amino acid. Okay, So when we do such a things, this is simply introduce this to 293 cells, and after selection of 2 days, and then we just induce the gene expression with the tetracycline and then monitor the phenotype of the cellular response. As you can see here, wild type replacement basically does not do anything. Okay, they grow equally well with or without induction or replacement. But when you have mutations introduced, then you use retarded growth. So it's very obvious phenotype at the cellular level using this experimental system. And then we apply the same strategy to other splicing factor like UTF-35, and you get exactly the same thing. Okay, but if you use the exact same strategy applied to mutation identify 65, there's no effect. And then actually this kind of a phenotypic assay is almost yes or no answer to those mutations. So in the other word, there are many mutations associated with disease. Does not necessarily mean every single one is a driver mutations. Okay, and the field, because they are statistically significant, everybody thinks all those mutations are driver event, which we disagree. And you will see every data is consistent with this initial growth analysis at the cellular levels. As I said, this is in 293 cells. You may argue this has nothing to do with blood disease or disorder. Now we do the same thing in this the only kind of a cellular model for MDS is there's cell lines derived from MDS patient, but does not carry any mutation in those genes. And if you do the same assay, you get exactly the same phenotype. And the same is true if you introduce mutations in mouse embryo fibroblast, which is normal cells, and you get the same phenotype. In other words, the repressed different mutations or retarded cell growth, although there are different degrees. Okay, so in this case, this is clearly that inhibiting cell growth by mutations in SR-SF2 and UTF-35, but not 65. So this is the first pass of a functional assay, clearly indicating which one is a driver and which mutation might be a passengers. Okay, and if you take those cells and go through this analysis of apoptosis or cell proliferation, which is normally people do, and we do the same thing. And you can see here that with the wild type before and after addition of DOX to induce, to cause a gene replacement, this cell cycle profiling remain identical. So the tetracycline does not do anything, but however, if you have introduced mutation, it causes clearly elevated G2M increase, the population. Okay, and UTF-35, you get exactly the same thing. Again, 65 mutations has nothing detectable. And then this, when you, this is done in all the cell types, they all show the same thing. And then this is on primary mouse embryo fibroblast. When you introduce mutations, we start to detect that there's a beta-gal activity, which is a sign of cell senescence. And you can see the quantitative level compared to a wild type, they all show a highly elevated degree of cell senescence. And just reminding you, cell senescence is kind of a cell do not grow kind of a phenotype, is a pre-oncogenic syndrome. So that has been established before a cell becomes fully transformed. So that's indicating this mutation has oncogenic potential once it coupled with something else. Okay, and of course in the field, the key question is what's something else? Remember, MDS has a 30% tendency to transient into full-blown leukemia like AML. Okay, so that's one of the long-term goals we still don't know yet. Of course, if it's splicing mutations, we have all those cellular models. We just take our normal technology going on in the lab for profiling alternative splicing. And then the data becomes quite clear when you put all those data together. You can see here, this is UTF-35. This is the cluster analysis with the one class of mutations. They all cluster together. And then this is another mutation in the same genes and across differently. So this data indicating even the mutation in the different mutation in the same gene cause a different splicing change. Okay, and then here is SR-SF2 all the mutations cause similar change because this is the same gene, but because the mutations are all happening in the single amino acid, change to single amino acid. Like the cellular assay or apoptosis cell cycle assay, we fail to record any phenotype with 65, and the same is true for the splicing response. Okay, and then the rest of them are all controls without adding tetracycline, they don't do anything. Okay, and if you pull those data together, it's clear that this mutant, all this mutant, we can record their splicing response largely cause induced exon skipping. Okay, but what's interesting here is that if you make a comparison for either skipped group or included group, there's no overlap, either between mutation between different splicing factors or different mutation in the same splicing factors. So this raises the questions: how altered splicing are responsible for disease? That if they cause a different change at a molecular level. So actually, this has been shown not only in our systems and also observed by others, and by sequencing the patient, by us and others, so that the whole field now is in kind of a confusion state. How can we deal with this kind of moving target that they change everything yet not identical and they cause the same disease phenotype? Of course, you can argue MDS is such a heterogeneous disease state that maybe is a mixed bag of different disease. But clearly, this raises questions whether or not we are all led to a wrong direction. Based on the rationale, splicing factor cause mutation by splicing defect. How do we know? Because in the, Amy just mentioned, there are a lot of genes has a multifunctional. And then how do we know assigned function was responsible for the phenotype? Okay, so in that regard, we need to be open-minded. That's the message I like to convey today. And we are part of the ENCODE project, just like non-hypothesis-driven, just chip your DNA, RNA binding protein on chromatin. And it turns out what we noticed that the most of the factors, about 60% of RNA binding proteins are chipped in specific regions in the chromatin, and most of them actually decorated on promoters as well as enhancer or other regulatory regions. And show here, this be fuzzy SRSF2 is like that, which is 35 and 30, UDF 35 and 65, and a bunch of SR protein like SRSF2 being representative one also showed a similar pattern. So that begins to give us some thought that it's not just splicing, but it's some other life of those factors at a genomic level that may be relevant to disease. For example, we published early on that the SR protein, this SRSF2, if you compare their binding on chromatin versus binding on RNA by clip or by chip, you can see they interact with a promoter at the DNA level, but they interact with exonic regions at the RNA levels. Okay, so this actually indicates that the transcriptional coupling at the promoter region transcription started with the joint venture of a splicing factor and the transcription factor at the promoter region, followed by the transition to the RNA level interaction at the later stage of splicing. And in fact, this image was published a long time ago in the Drosophila system. This is corresponding to the homolog of this guy in Drosophila. Actually, they looks like a hot dog that sandwiched the PO2 in the middle. So they may trap those things, travel these things together on this induced heat shock locus. Okay, in that rationale, we recall our early literature, and all of a sudden we realize there are another mechanisms involved, which is so-called this phenomenon of R-loop formation. So what's the R-loop? Is that when PO2 going through transcription, moving along the DNA, that they produce nascent RNA. Nascent RNA come out from RNA channel, which are separated from the template DNA. But however, and normally this RNA will go through initial binding by RNA binding protein, which commit them to the splicing pathway. Now they are released from the template region. Okay, so as a result, this transcriptional bubble remain dynamic and small when the polymerase is moving along the gene or after promoters. But however, and if this RNA do not get removed, they can yield back at the back because there was this transcriptional bubble with open region and has extensive kind of annealing. And this actually create retarding force to slow down polymerase. Okay, this R loop formation. And in the meantime, this exposure of this excessive R loop expose single-stranded DNA, not invite attack by all kind of DNA damage reagent, not get linked to DNA damage. So all those kind of things are happening at this interface of transcription and co-transcriptional splicing. And then we know SR proteins, there's splicing factors that involve in 3' splice site definition, all cause these kind of things based on the study initially published by Jim Manning group and later on published by us. And indeed, we found when you have mutations and you all cause DNA damage as indicated by this gamma H2X staining and as quantified here, okay? And it also cause replication stress when you label replicate DNA with 2 different dyne, then you can count the lengths, and then with the replacement with expression of mutant, then this length gets shortened as indicated here. Okay, this is different mutations in different factors. So that indicating that the genome instability is associated with these mutations. But this is in conflict with the general notion that leukemia has the lowest mutation rate. How can we put these 2 puzzles together? No mutation rate yet induced the genome instability. So we checked the pathway invoked, and it turns out that you see, when you detect the gamma H2X at the biochemical level, they clearly induced by the mutations, which is true regardless of the mutation in different splicing factors. And the PP3, phosphorylated PP3 at the serine 15 level show the same thing. And then we check 1, which is indicator for the activation of ATR pathway, which is associated with a single-strand DNA break, and clearly they get activated. And in comparison, the same study showed check 2 has low effect. This is responsible for activation of ATM pathway associated with a double-strand break. So in the other word, splicing factor mutation insult the genome by causing single-strand break exclusively. That explains that even though you started with single-strand break, if it's very excessive, then you convert it doing replication into double-strand break that activate ATM, so that leads to high mutation rate, such as what you see in cancer. And then in contrast, and then you get repaired, and then you just transiently actively check 1. As a result, you lead to cell cycle arrest, but not high mutation rate. And the question is, how can you detect such an R loop? We detected a new technology to use catalytic inactivated RNase H, not binded to DNA RNA hybrid in vivo. And so that you can using these mutations. And also we have a control mutation that has a binding site mutation that they're not bind. And using this, and you can see that a mutant can induce R loop formation and a wild type don't. And a different mutation all significantly induced dynamic R loop formation in vivo. So, and then whether or not this is relevant for disease, now we can use the active RNase H to quench the R loop. You can degrade the RNA, and you can see once you have this RNase H wild type, then you can get rid of this activation. Okay, so that means only active but not binding mutant RNase H is able to rescue the growth phenotype. And finally, this is the final slide. I'm done. And then we apply this to at the cellular level, and you can see here that all those mutations, if it's the binding site mutations, RNase H, there's no rescue. And if it's the mutant, if it's a wild type of RNase H, and you can see here, once you induce their expression, it rescued from this to this level. It's not a complete rescue, but a partial rescue. And at the DNA damage level, clearly it's rescued very satisfactorily. And we can also use in knock-in mice, which show the plating efficiency. This is the proliferation potential of early progenitor cells is reduced, but once you express RNase H, then it can restore partially its functions. So the take-home message from all the studies is splicing factor mutations cause extensive but long-overlapping change in splicing. They suggest a contribution but not causal to the MDS etiology. Instead, the causal mutation in splicing factor induces single-strand but not double-strand break, which lead to the activation of ATR but not ATM pathway, and this provided a mechanistic insight into the induced genome instability by the low mutation rate in leukemia. And the induced R loop seems to be underlying the genome instability induced, indicating defect at the very beginning of a transcriptional splicing as the cause of the as the mechanistic and count mechanistically to the phenotype. And finally, RNase H is able to functionally rescue the compromised hemopoietic potential of early blood progenitor cells. This demonstrates the causal effect of excessive R loop formation to the disease phenotype. Finally, I'd like to acknowledge my colleague, not only this Liang Chen, a postdoc, and Jerry Chen. This is experimental postdoc, is the informatics postdoc in the lab, in collaboration, close collaboration with the Dong Zhang lab in study this disease mechanism. Thank you for your attention.
That was great. Thanks very much. We have time for a few questions.
Hi, I was wondering, you said it wasn't causal, but when you look at the high-risk MDS patients, there's two questions. Have you analyzed them to see if they have a higher rate of these splice mutations? And secondly,
say it again. Let me make sure. Yeah.
When you have there's intermediate and high-risk MDS. In other words, MDS patients that are staged to be imminent to shift to the AML phenotype of subset.
Correct. Yeah.
Have you looked at them to see if there's a correlation between those patients and the degree of mutations you see, splicing mutations you see?
Yeah, we just had a paper publishing RNA. They do show that the mutations. And it turns out it's the same thing in patient, that the different mutations, mutation in different splicing factor was different mutation in the same splicing factor cause different splicing change. And they are not related, much related to the degree. Certainly not related to early MDS phenotype versus AML, not advanced to AML. In fact, this R loop may be responsible because this initial self-senescence phenotype can be fully rescued if you have a mutation in PP3 pathway. So in the other word, a lot of secondary things happening that overcome such an initial response that may be responsible for the disease advance.
Very good. And the second question, and I'll admit it's a bias of mine, would these R-loop effectors possibly affect the T-loop, and therefore would you be collecting or switching on telomerase or affecting telomeres? Because it's thought that part of the obligate flip to the immortal AML cell is telomerase activation, which can be somewhat quiescent in subsets of the MDS. Have you looked at that?
I have not, but it has been done by what is his name? I mean, this guy from MD Anderson published last year in Cancer Cell that with reactivated telomerase that created genome instability that actually gave rise to this kind of MDS-like phenotype in mice.
Yeah, I thought the R-loop work was great. So I had a question. When you have the R-loop and then you have the DNA strand, you have the RNA that could be edited, but the DNA changes that we see in clonal hematopoiesis and sometimes in high-risk MDS are C to T changes. And I was thinking, have you looked at ApoBec as opposed to the standard, genomic instability mechanisms as an enzymatic source of mutagenesis?
That could be true because this ApoBec protein is involved in this kind of thing, especially sometimes this triggered translocation. That's a different game. But this R loop, give us some hint. In fact, I just utilize this time to extend this R loop business. The entire field has been using an antibody called S6.9 or 9.6 or whatever. That antibody is specifically recognized RNA-DNA hybrid. So people have been isolating genomic DNA and using this antibody to capture. And actually, that's all everybody is doing. But the problem is that this is very sensitive to the isolation procedure because you can lose many of those. And then this one is captured in vivo. Turns out R loop is more dynamic than you thought. And then that caused a lot of changes. Yeah.